What is the optimal temperature of thermophiles? How about mesophiles? What is superoxide dismutase? Can you easily answer those questions? If not, continue watching my video. I'll answer them and more. I will be breaking down microbiology questions so that you can do well on your exam. Using the following information to answer questions 1 and 2, two culture media were incubated with 4 different bacteria. After incubation, the following results were obtained, and you see that on the screen. Medium 1 has red colonies, no growth, colorless colonies. Medium 2 has no growth, growth, and no growth. Let's go to number 1. Media number 1. Was selective media used, differential, or selective and differential? Well, what is selective media? Well, it is used when you want to suppress growth of unwanted bacteria and encourage growth of desired bacteria. Differential media is used when you want to distinguish between the colonies of your distinguish the colonies of your desired organism from other colonies growing on the same plate. Medium 1. You have red colonies, no growth, and colorless colonies. Here you see a distinguish between the colonies, but you also see that there is growth and no growth. Now, going back to our definitions, we see that medium 1 is selective and differential. So the answer to number 1 is C. Number 2, medium 2, you see no growth, no growth. We have selective for A, B, differential, C, both selective and differential. Well, in medium 2, you don't see a distinguish of colonies like you do in medium 1. So we can eliminate B and we can eliminate C, which leaves us with A of being the answer. And it's A because there is growth that was suppressed, or there wasn't any growth, so it was suppressed, but there was growth in the other 2 of the desired bacteria. So it is selective. Let's go to number 3 and number 4. We're going to use this chart. Number 3. Which of the lines best depicts the log phase of the thermophile incubated at room temperature? So above we have this chart where on the x-axis we have time, and on the y we have the log 10 of the number of cells. Thermophile, what is it? Well, it is a heat-loving microorganism. So we have heat-loving microbe. Its optimum growth is between 50 to 60 degrees Celsius. We also know in the question that it's at room temperature, and the room temperature is between 20 to 23 degrees Celsius. So does this temperature, room temperature, fit the optimum growth for a thermophile? Well, it doesn't. And since the optimal growth is not met at this room temperature, we're going to see no growth. And on the chart, where would you see a no increase in the number of cells? A, B, or C? Well, the answer is C. Number 4. Which of the lines best depicts the log phase of Listeria monocytogenes growing in humans? So, Listeria is a mesophile. And its optimal temperature is 25 degrees to 40 degrees Celsius. And it's saying it's growing in humans. The temperature of humans is 37 degrees Celsius. So does 37 degrees fit into the optimal temperature range of mesophiles? And I spelled it wrong. I don't know why I put like a little cross over it. And it does. So that's a yes. Going back to the chart, since there's optimal growth, we're going to see a huge increase in the number of cells. So which line on the graph shows a major increase in the number of cells? Well, it's A. Let's move this up. Number 5. 5. Assume you inoculated 100 facultative anaerobic cells onto nutrient agar and incubated the plate aerobically. Then you inoculated 100 cells of the same species onto nutrient agar and incubated the second plate anaerobically. After incubation for 24 hours, you should have A. More colonies on the aerobic plate. B. More colonies on the anaerobic plate. C. The same number of colonies on both plates. Well, we need to know what a facultative anaerobic cell is. Well, it is a microbe that can use oxygen, so it can use oxygen when present, and it can also grow without oxygen. In an aerobic situation, anaerobic, respiration, and in fermentation. Okay, so in this situation, one was placed or incubated in an aerobic situation. Let's erase that. And the other was incubated in an anaerobic situation. Now, more colonies on the aerobic plate. You would see more? Yeah, you would see more because they can grow in the presence of oxygen. More colonies on the anaerobic plate? Well, yeah, there'll be more colonies on the anaerobic plate because they can grow without oxygen in anaerobic respiration and fermentation. So that's true. You'll receive the same number of colonies on both plates. Since you see growth in both anaerobic and aerobic, you'll see the same number of colonies on both plates. So the answer is C. 6. The term trace elements refers to A. the elements CHONPS, B. vitamins, C. nitrogen, phosphorus, and sulfur, D. small mineral requirements, and E. toxic substance. In order to answer this question, you need to know what trace elements are. Well, trace elements are small amounts of minerals that are required such as iron, copper, and zinc. I'll give you some examples. Iron, copper, and zinc. So now that we know the definition of what trace elements are and we have some examples, we can go back to our options. A, the elements CHONPS, that's carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And since that's not one of our, the examples that I gave, it cannot be A. And so we can go down to C, nitrogen, phosphorus, and sulfur, which is in CHONPS. We can cancel that one out. We know it's not toxic, so we can cancel that one out. We're left with B and D, vitamins and small mineral requirements. Well, going back to our definition, there's small amounts of minerals that are required, and D matches our definition. So the answer is D. Question number 7. Which one of the following temperatures would most likely kill a mesophile? It's very important to pay close attention to your questions. It says which one would most likely kill a mesophile. Well, what is a mesophile? Well, the optimal temperature of a mesophile is 25 degrees to 40 degrees Celsius. That's its optimal growth. We want to know outside of this temperature, which one would kill a mesophile? We have A, minus 50 degrees. It could kill it, but most likely it would stop growth or slow it down. So it would not be minus 50. 0 degrees would slow it down, slow down the growth. 9 degrees would slow down the growth. 57 will fit right in the range of its optimal, so it would not kill it. And then we're left with 60 degrees, which would be the same or optimal temperature of a thermophile. And we are talking about mesophile, so it's outside of its optimal growth and can kill. So the answer is E. Number 8. Which of the following is not a characteristic of biofilms? A, antibiotic resistance. Well, that is true. It is a characteristic because it has a slime layer that protects the bacterium that are inside. We have B, a hydrogel. Well, hydrogel is a complex polymer. That's associated with biofilms. So this is a true. Iron deficiency. As a lack of iron inhibits surface motility. It helps aggregate bacteria, which would give us a biofilm. So we're going to put lack of iron inhibits surface motility. And then D, quorum sensing. It is a way that they communicate. So it's cell to cell chemical communication. And Number 8. Which of the following is not a characteristic of biofilms? A, antibiotic resistance. Well, that is true. It is a characteristic because it has a slime layer that protects the bacterium that are inside. We have B, a hydrogel. Well, hydrogel is a complex polymer. that's associated with biofilms. So this is a true. Iron deficiency. As a lack of iron inhibits surface motility, it helps aggregate bacteria, which would give us a biofilm. So we're going to put lack of iron inhibits surface motility. And then D, quorum sensing. It is a way that they communicate. So it's cell to cell chemical communication. And they have that, so that's true. What we're left with is C. So the answer is C. Lack of iron inhibits the surface motility. This helps, the iron helps to aggregate the bacteria and create the biofilm. Number 9. Which of the following types of media would not be used to culture aerobes? Best to answer this question is to know what selective, reducing, enrichment, and differential and complex media are. Well, selective, we've gone through what that is. It is to suppress growth of unwanted bacteria and encourage growth of desired microbes. And I hope you're taking notes. We have D, which is differential. We went over that one. And it's the distinguished colonies of desired organisms from other colonies growing in the same plate. So now that we know those two, we're going to go to the ones that we do not know. Reducing media. It depletes oxygen in culture medium by using sodium thioglycolate with dissolved oxygen. Enriched media. Well, it's liquid and there are nutrients and the environment conditions should be right for growth. So environment, let's put some space here so you can tell that it says environment and not 10. Environment conditions for growth of a particular microbe. But others, but not others. There is an increase in very small number of organisms to detectable levels. Oops. If I can spell, let's go back one and then put levels. All right. Complex media. Complex media they have nutrients plus extracts from yeast, meats, and plants. Proteins give the bacteria energy, carbon, nitrogen, and sulfur. So we have our definitions. We know what selective is from our previous question. Reducing, it depletes oxygen in culture media by using sodium thioglycolate with dissolved oxygen. Enrichment media, it's liquid. It has nutrients and the environment conditions for growth for a particular microbe, but not others. There's an increase in very small numbers of organisms to the detectable levels. We know differential media from our previous question. Again, hope you're taking notes. And E, complex media, nutrients and extracts from yeast and meats and plants. Proteins give bacteria energy, carbon, nitrogen, and sulfur. So which of the following best types of media would not be used to culture aerobes? Well, the answer would be B, because it depletes oxygen. And aerobes, they need oxygen, so they grow in oxygen. And we do not need a media that depletes oxygen. So the answer is B. Number 10, our last one. An organism that has peroxidase and superoxide dismutase but lacks catalase is most likely an aerobe, aerotolerant anaerobe, obligate anaerobe. Well, what is peroxidase? What is superoxide dismutase? And what is catalase? Let's get these definitions. So we have peroxidase. I'm just put P for short. It breaks down hydrogen peroxide but doesn't produce oxygen. But produces water. We have superoxide dismutase. I'm gonna put SD. And it breaks down, well, its abbreviation's SOD. That's what you would see in your textbook. And so it's an enzyme that breaks down or neutralizes toxic superoxide radicals. O2 negative. And then we have catalase. I'll just put C. And catalase breaks down hydrogen peroxide and produces water and oxygen. So we know what these enzymes, what they do. Let's go to our options. Well, aerobes, they use oxygen for growth. B, aerotolerant anaerobes, they cannot use oxygen. But they can tolerate it. And obligate anaerobes, they are unable to use molecular oxygen for energy yielding reactions. And oxygen kills them. All right, now that we have all of our definitions down, we know what's what, let's go back to our question, our statement. It's an organism that has peroxidase and superoxide dismutase but lacks catalase is most likely, well, in respiration, radicals are produced. And we know here that SODs neutralizes them and produces H2O2, hydrogen peroxide, and O2. Well, we go back to our options. We know that our answer cannot be C because they are obligate anaerobes are killed in the presence of oxygen, and oxygen is present. Next one, we have peroxidase and entered into the system or created. Peroxidase. And what they do is they break down hydrogen peroxide to give H2O, but no oxygen. But we need oxygen for anaerobes, so the answer would not be A. What we're left with is B, which is the correct answer. Now, it cannot use the oxygen, but it can tolerate it, and oxygen is present here, and so aerotolerant anaerobes can tolerate the oxygen, but it just won't be used by them. Was this video helpful? Then go ahead and do that YouTube stuff, that the like, comment, share, and don't forget to subscribe. Also, in my description box, you'll be able to join my micro tribe where I have more questions that I break down to help you on your next exam. All right, see you there.